Welcome to the Material Minds podcast, where we talk to the people exploring new materials for a better world. Today we talked with Sara Trujillo, the head of the Materials Host Interactions Research Group here at INM. We talked about cells in the body, whether they're alive or dead. We talked about a cat named Fluffy, whose bone she helped regenerate. And we also talked about the current research that she's doing here about how to make new materials that are safe and compatible within the body. We also finally talked about her favorite material, which allows her to combine science and art. So I hope you enjoy the podcast, and here we go. It's great to have you here, Sara.
Hi, nice to be here.
Yeah, so I introduce you very briefly to our listeners, and so we want to get to know more of you, but through you, with the game of 3 truths and a lie, where you tell me 4 statements, and one of them is a carefully crafted lie, and I'm going to try to guess which one that is.
Okay.
So if you're ready,
Sounds
just go
good.
ahead.
Yeah, I've been working on this for some time.
All right.
I'm a very bad liar, so I'm sure you're going to pick me.
I'm not good at reading faces, so I've got to only work on logic. Yeah.
All right. So my first statement is that this is not the first podcast I've been invited as a guest.
Mm-hmm.
Although we didn't have this fancy, setup. So this is
your
my
first.
thing.
Okay. I'm going to ask you some questions to try to dig into the truth. So what was the podcast about?
So the podcast was about science as well. So I was there as a guest, as a postdoctoral researcher in my former group. And we were just talking about our science and kind of doing some sort of public engagement, science communication kind of activities with the students. And yeah, it was a great time.
Yeah, and was it uploaded? Is it available even now?
It is available
it's
on your favorite podcast app.
On Spotify?
Yeah, you can.
Okay,
find it
and on
Spotify.
other platforms, I suppose.
Yeah.
Okay. How long was the podcast for?
Oh, we actually had to record twice because the first time we had some technical problems with the microphones and the sound. And we did record a second time. And I think that at the end, the length was around 30 minutes, 40 maybe.
Okay.
Yeah.
Okay, I think that's a good amount of information that I have now, but yeah, it's very difficult to guess.
Okay.
So let's move on to your next one, and then
Perfect.
maybe that helps me.
So the second statement that I wrote is actually a fun fact. So I don't know if you know that in the human body, there are 300 times more cells than there are stars in the Milky Way.
Okay.
What do you think about this?
Well, I looked up just yesterday that the Milky Way has 100 billion stars.
All right.
So you said 360 times.
I said around 300 times.
Around
more,
300 times.
without counting the bacteria.
Without counting the bacteria.
Yeah.
Okay. Why do you know this fact? Or did you look it up for this game particularly?
I kind of looked for the numbers, but
Yeah.
I did know that we have lots of cells. And that I'm a bit nerdy on this kind of fact number. So I wanted to bring something around that here today.
Yeah.
So, yeah.
Okay. I did not anytime recently or even in the past really look up how many cells we have in the body. So I'll have to do a lot of heavy calculation to figure this out. But maybe we'll go to your next statement.
Would you have a guess on how many cells you have? Or
I have a feeling like 30 trillion could be a number that I've seen somewhere.
okay.
But I'm not entirely sure about that. So that's why I think it could be true.
Okay.
And that is 300 times more than 100 billion. So, okay.
Yeah.
The thing is, I don't know if it's what I saw was maybe I remember 30 something because I've looked up how many bacteria are there. And there used to be this kind of, like false information that there are 10 times more bacteria than human cells
Yeah.
in the body.
Yeah.
And then it was discovered that this was just the back of envelope calculation by someone back in the day based on the weight of the microbiome and the size of a bacteria. But more accurate results have shown that you have about the same number in the end, or maybe even a bit more mammalian cells or human cells than the bacteria. So I feel in all of those things that I read 30 illion something. So I don't know if it's trillion or billion, and then this is where I'm a little bit lost. But let's go to your third statement and see if we get
I
there.
don't want to give away too much.
Okay.
Okay, so my third statement is that a cell can actually be dead and alive at the same time.
Dead and alive at the same time. Okay, so what is this something you have experienced in your work?
Yes, actually, because sometimes, you look at one experiment and you see, all right, these cells have the cell membrane intact, but I do see that there's very little metabolic activity on a different experiment. Or maybe I do a staining and I have both stainings at the same time happening. So the dead staining is showing up, but the cell is also metabolizing the live staining.
Okay.
So it's kind of contradictory sometimes, the results you get. So yeah, that's why.
So is the definition of dead and live based on the measurement you're doing? Like that you use the stain and it tells you whether it's dead or alive, and then you use a different measurement of metabolic activity, and then that tells you another result.
Yeah, so I mean, that's how scientists usually work, no? They look for some answers and you usually don't get the full picture, right? So you ask one question and you get the answer to this question.
So for this statement, what is your definition of live and dead for a cell?
That is a good question. So I would say that my definition of a live cell would be a cell that's able to have some metabolic activity and have at least two of the main membranes intact.
Okay. And okay, so that's a live cell.
Yeah.
Yeah.
That could
okay.
be a live cell.
In this.
We can. Someone could argue.
Oh yeah. It's a deep philosophical debate. Once you go down to single cells and bacteria and viruses and so on. Yeah.
Yes.
It's a fine line between the two. But okay, I'll think about this. Let's go with your fourth statement.
Okay. My fourth statement is about the work that I've done in the past. So I once worked for a project that its aim was to help landmine victims. So to reconstruct large pieces of bone because they had lost some extremities. And one of the first patients that we tested with our technology was a cat named Fluffy.
A cat named Fluffy. So you tested bone regeneration.
Correct.
On a cat.
Correct.
Named Fluffy. Okay.
Named Fluffy. The devil is in the details.
Yeah. I mean, if it's a lie, it's very well crafted. So, okay. So how does this bone regeneration work, and why did you test in a cat and not like a mouse or whatever?
Okay. So in the lab I was working in the past, we have this technology where we use a protein that's found normally in the extracellular matrix. And this protein has the ability to bind different growth factors. So growth factors are these molecules that can trigger different differentiation pathways. So the beauty of the protein is that it can bind these factors together with cell binding. So it kind of maximizes the exposure of the cell to this factor.
So it allows the cell to stick to your material
Yeah.
And it gives
see
it
this
the signal.
molecule, yeah,
that
very efficiently.
Okay.
So the idea is
okay.
that we can use this growth factor delivery with very low amounts.
Uh-huh.
And yeah, the research was in the direction of how to make this connection into a biomaterial scaffold or yeah.
Okay, so so your material is basically, it's a scaffold that you put on a broken bone, and then the material will allow the cells to stick to it,
Yeah.
and will signal it to try to
Yes.
grow faster. And so by itself, the bone will not be able to grow that fast.
Yeah, so the idea is
de-heal.
to use this for large pieces of bone. So if you have a fracture that's big enough, your body cannot regenerate it.
Yeah.
So the idea was to use this extra help to kind of bridge the gap.
Is that why you went for a cat instead of a mouse, or did you already do mouse studies and
We
then
already
go to
did
a
my studies.
Yeah,
And
okay.
I said this was one of the first patients.
This was the patient. Yeah,
Yeah.
okay, that's a good point. Yeah, uh-huh.
So yeah, these were pets that we were able to treat.
Okay.
And this was kind of one of the first steps to go into human, which is
Okay.
still ongoing.
Yeah. Okay. Shall we go through your 4 statements again?
Yeah, let's do
I
it.
have a refresher, and then I can
Okay.
think it over.
So first, this is not my first
Yeah.
podcast.
Okay.
The second is that there are 300 times more human cells in the body than stars are in the Milky Way.
Yeah.
The third statement was that a cell can at the same time be dead and alive.
Yeah.
And the fourth one is that I worked in a project to treat bone fractures or large pieces of bone in landmine victims.
Okay. And in the last statement, the lie could not be part of, like, the cat's name was not Fluffy or something like that, right?
No.
Yeah. Okay. So it's
that
about
would-
the project.
That would
Yeah.
have been
Yeah. Yeah. I don't know how you lie. So, the thing is, the first one, I have no read on that, because it's something that you could have obviously. Okay, you said it was in your postdoc when you were probably doing the bone regeneration stuff. So was the podcast about that science that you were doing?
Yeah, it was.
Okay, and was it about the cat, or was it after the cat?
No, it was before the cat.
It was before the cat.
Cat
Okay.
happened after.
Okay. I keep getting back to the second statement.
Okay.
That is 300 times more cells. But now that you made me think about it, I have this number of 30 trillion stuck in my head, and that it could be possible. That could be possible. I'm okay. I've got to make a decision now. So podcast, cell can be live and dead. Live and dead seems like something reasonable to me, and then bone regeneration. And bone regeneration seems like the kind of research you would have done in your biomaterials work. I want to go with statement number 2. Is
The
that
number of
right?
cells?
Number of cells?
That is not right.
That's not right?
You were actually...
I was right with the numbers?
You were right with the numbers. I was, how can he know?
Yeah, I read a book long ago about the microbiome, and again,
Okay,
so I'm
cool,
interested in
yeah.
the bacteria,
Yeah.
and so I got the numbers from there, I think. But I was saying, like, 30 trillion seems like the number I'm thinking of, but it just doesn't seem right, I felt. But okay, that is right. Then I lost the game. Is it the first one?
No, that was
No?
true. Also.
That was true?
It was
Yeah.
actually very difficult
I'm
for
so
me to
bad.
find a lie.
Yeah.
So I kind of went the philosophical route for
Okay,
it.
the live and dead?
Yeah.
Oh.
So you know
Yeah.
that scientists, we do love to classify things and to say
Yeah.
this is one thing, this is another thing. So there's actually a definition of what a life and dead
Okay.
cell look like.
Uh-huh.
And there is a point of no return, so to
Okay.
speak, where you can see that the cell is dead because there's no reversibility to it.
Okay, so even if the membrane, let's say, is intact, or let's say the membrane's not so intact, but the metabolism's kind of running, it could still be a point of no return that,
Yeah.
then you consider it dead,
Yeah, correct.
basically. Yeah.
Yeah.
Okay. Okay, that was very sneaky. Like,
I mean,
because I
yeah,
asked you for the
I had to
definition.
do
Yeah, yeah.
something.
No,
Like,
no,
how
that
do
was
you
that was
lie?
really good. That was really good. No, that was really good. And I would not have guessed that one, and
Okay.
I did not guess
I'm
that
glad
one.
that I kind of won the game there, right?
Well, it shows that you're a better liar than you thought,
Yeah,
right? Yeah,
I thought it was
yeah.
gonna be tough. I'm actually like very tense with the lying, so I don't know if you can tell. But
No, I feel like your facial expression was similar for all the statements.
I do like playing poker from time to
Yeah,
time. So
oh
you
man,
know.
I would not want to play with you. I think that's not a game I'm going to win. But okay, that's fascinating. That's really interesting. Is that related to your research, that statement itself, like where it's important for you to know that a cell is alive or dead?
Yes, actually, yes. At our group, in the materials host interactions group, we look at how the body reacts to different biomaterials. And toxicity is a big part of it, right? Because you have these very new fancy polymers that are synthesized, and there's always things that can interact with in different manners with the cells. And it's actually one of the first things we looked at.
Okay.
Is how's the toxicity of this material? And yeah, we try to kind of ask the same questions on different ways. So we have the full picture of these things and also, different time points and yeah.
Okay. And then so to do the right measurement to know whether you've hit this point of no return, kind of to classify?
I mean, yeah, it depends on the answers we get from the first experiments, if we need to have the extra confirmation or if we need more time to see if this reversibility is still ongoing. We
Okay,
do
fascinating.
these
Yeah.
things.
I'm gonna actually scoot back to the podcast thing.
Yeah, cool.
Like how did that happen? And if it was not about the cat, what was it about? Because I want to get to the cat also again.
Okay.
Yeah.
Yeah, so this was an initiative from the students.
Uh-huh.
So we had a few of different science communication activities. And there were 2 PhD students that took on the leading role of starting a podcast. They did everything on the podcast, like they hosted it, they prepared it, they edited it, and everything.
Yeah.
And yeah, I was super glad that they chose me as one of the guests. So I was very happy to be, helping.
Yeah.
So that was quite a nice experience also to see the students kind of get in their role of hosts and kind of thinking of ways to engage with people and with me as a guest. That was
That's really great. And
good.
what students were these? PhDs or masters?
Yes, they were PhD students.
PhD students.
Yeah.
Okay. And this was their own initiative that they wanted to have the science out there?
Yeah,
Okay.
yeah, definitely.
Okay, that's very interesting. How long was that podcast?
Oh, I think it was like 3 or 4 episodes.
3 or 4 episodes. Yeah. Okay. But with you.
No, with
okay,
me,
okay.
just one, obviously.
And that was like how long was the conversation there?
It was around 30 to 40 minutes.
Okay. Nice.
Yeah, it was pretty good.
Yeah. And so which point of research were you on regarding this bone regen in a cat? Like were you with mice or had you just started when you were doing that podcast?
Yeah, so when I was doing the podcast, I was working with a similar strategy, but in a different scaffold material. So I was working with hydrogels. And I was using this protein that, combine these growth factors into a hydrogel system. So I was talking a little bit about my findings on how I could incorporate the protein into more a better mimic of the extracellular matrix that we have in the body, which is through a hydrogel. So hydrogels are these polymeric materials that contain lots of water, and then you can tweak them and tune them to also match different physicochemical properties of the body. So they're very useful in tissue regeneration and other fields.
Yeah.
I was talking about how I reached the point of having this protein into this hydrogel system, and how I was, looking at growth factor delivery with this.
Yeah, it's very interesting. Is it something? Maybe you can. That's a good segue to get back into like how you got there. Is this something that you had in mind since your undergraduate and graduate times, or is it something that happened over time? How did that evolve?
Yeah, that's a good question. So I've always been interested in cell biology and cell therapy. So I came across tissue engineering just by chance. At the university I was studying in Spain, in Valencia, there was a center there that were working on tissue regeneration. And I kind of started as a bachelor student doing my bachelor thesis.
Okay.
I continued as a master student. So there I was working on bone regeneration with a different approach. So we used these plastic scaffolds. So there are some thermoplastics that you can shape and make them porous, for example, to allow the cells to go in. So we were trying to reinforce these plastic scaffolds with bioactive glass.
Okay.
I don't know if you've heard of bioactive glass.
Not much.
So bioactive glass are inorganic glasses.
Okay.
So they're made of calcium, silicon,
okay.
all these things.
Okay.
And the good thing about these glasses is that they're hard. So that's why we use them to reinforce the polymer.
Yeah.
But they're super brittle.
Okay.
So then you need to have both components.
Sorry, but why are they bioactive?
They're bioactive because they can trigger mineralization spontaneously.
Okay. So then you have the hardness of the bone coming in.
Yeah, correct. So the mineral part of the bone kind of deposits there in the right conformation. So form these very beautiful cauliflowers.
Okay. Yeah.
And it's really cool to see in
the calcium crystals, kind of that form out of it. Does the glass itself kind of biodegrade and release the minerals?
Yes.
Okay.
With time.
And also the thermoplastic that you had, is that also something that
so at the time I was working with polycaprolactone and polylactic acid, which are degradable, long-term degradability. But so the idea was kind of use it as a first entry point for the bone to grow, and then with time to degrade and disappear.
Okay. And is this something that you said you did through your bachelor and master? Is it something you continued through your PhD?
So the bone topic, yes, because my PhD was about these hydrogels that in the end were used for bone regeneration and also for angiogenesis. That was like a nice extra.
Nice. Yeah. Angiogenesis meaning blood vessel regeneration.
Yeah, correct. Capillary,
right?
new capillary formation. Yeah.
New capillary formation. Yeah. Okay.
So I kind of wanted to follow on the topic of bone regeneration, and that is why I kind of moved away from Valencia, and I went to Scotland to pursue my PhD.
Yeah, okay.
And I think that the hydrogel take was just a radical change of polymer for me, because it had very different properties to what I was used with these plastics. But it was super fun, and I enjoyed it a lot. That's why
really great.
I'm still a scientist, I guess.
Yeah. So through your PhD, you did the bone research, and you took that forward into your postdoc and got it all the way to a cat.
Yes, correct.
And what is the story of the cat?
Yeah, so for the cat, this was a short but very rewarding experience. So as a postdoc, I ended up in a project that was using this technology. Not with the hydrogels, that was what I developed during my PhD, but they use this protein on polymer coating. And the idea was to coat with this polymer bone chips, commercial bone chips.
Yeah.
And with that, triggered the regeneration on different veterinary patients. So we went with the veterinary patient route because of the accessibility. So we're very close to the veterinary hospital, and the veterinaries there were amazing.
Yeah.
They were super interested in the research. Also, because we were treating, how to say, like lost cases. So these were pets that had a very big fracture that the normal methods wouldn't help.
Yeah.
So the only other option was to amputate.
Okay.
So then what we did in this project was to offer this solution as a experimental research
Nice. Yeah.
solution, and then it was up to the owner to go through the extra surgery for
Yeah.
their pet or not. So yeah, that was as I said, super rewarding because
Yeah.
we were able to treat 9 dogs and 2 cats.
Yeah.
Fluffy was one that I remember very well because I attended the whole surgery with the. I mean, I didn't touch the animal,
okay.
obviously, but I did prepare some of the materials for the surgeries for that.
So you prepared the materials that went into the animal and healed her.
Yeah.
And now Fluffy's got a healed bone and is jumping and running around, or how strong is that bone? Is it fine?
I mean, depends on the pet and the fracture that they had, because some of the fractures were really tough.
Okay.
But I have to say that all of them are using the limbs. I mean,
So you're saying all of them, meaning there were multiple animals that were treated?
Yeah,
also?
So we treated 9
9
dogs and 2 cats.
9 dogs and 2 cats?
Yeah.
Okay, wow. That's amazing. Yeah.
Yeah,
Okay.
it was really good. Also, we received some videos of the pets just walking around just to show that they were, using the paws and so on. And yeah, it was a very nice experience because it was the first time that I could see, the applicability of the science that we were doing. So that was that was amazing, because sometimes you get lost in the lab, and you think of the big picture, but then seeing it happen in real life is
it's
really
extremely
nice.
rewarding. Yeah. So how far out do you think that is for humans now? Are human kind of trials starting off, or where are we?
So there's a thought on going into human. I actually have to double check with how the research is going, because I know that it's ongoing for sure.
Yeah.
So this is something that my former lab is pursuing.
At the group of Professor Manuel Salmon Sanchez. Yeah.
Yes, correct. But I haven't heard that there's any human trial yet.
Oh,
So
that would be
let's
amazing
see.
when this comes to humans. Eventually.
Yeah. I mean, the cool idea for the landmine victims was to kind of heal enough bone for them to be able to use a prosthetic, actually. Because sometimes they cannot get these prosthetics.
Yeah, okay.
Just because they don't have enough tissue there.
Okay, so they've lost their arm or whatever, limb.
Yeah.
And you just want to heal enough bone so that the prosthetic can be...
Yeah.
Oh, wow. This research is so deep and interesting. That's really nice. But talking about rewarding things, like also here at INM, your research is very close to application still, right? Like you want to study whether these biomaterials are compatible within the host, which is humans largely, I guess. But
Yes,
I guess
correct.
you're starting with animals.
Probably not pets.
Yeah.
Soon, at least. Yeah, exactly. So one of my main drives in science is the translational part, the applicability. I guess because of my past experiences. But yeah, I look forward to kind of help with all these new materials, these really crazy, amazing living materials that we make here at INM, and try to help bridge the gap into what you can do as a proof of concept material, which are all really nice ideas, and what it can actually be used in the body. So part of the research that we do at the group is not only to see what are the effects that what do the cells do when they see these materials, but also trying to help redesign these living materials into a better, more compatible device, so to speak. So that's I think it's a pretty cool topic. So we kind of get to see both worlds, like the designing of the material and kind of like growing the material into a fully device, and then testing it and seeing, okay, this has an actual application. This can do something important, no, for people.
And is this what your recent grant from the German Science Foundation from the DFG is about? Is it what it's allowing you to do?
Yes, exactly. So now we have this DFG grant that's about looking at biocompatibility of living materials. So our aim is to see what's the inflammatory response, for example, on materials that are meant to be implanted. So the idea is to see if these living materials are alive once you put them inside the body, not only on the outer part. Like most of the things you first think of is skin application, for example. But here we wanted to go one step further and try to see what's happening when you have these things in.
And just for our listeners who might not be aware of living materials, can you give your
I can give my take, of
description
course.
of it? Yeah, your take on it.
In front of
Well, we do work on that in our group also, but I'm very curious to hear from your perspective.
Yeah, so when I say living material, I'm referring to a hybrid system, so with two components. First one is a living microorganism. So in our case, we work with bacteria. So these are usually genetically modified bacteria that you know quite a lot about. And the idea is to have these bacteria secreting different therapeutics for drug delivery. And then the second component is a polymeric matrix. And the combination of both can give you a biomaterial that can do more than the two separate components by themselves. So the polymeric matrix is supposed to enclose these bacteria, protect them, but also protect the host from the bacteria themselves, and also allow the viability of these bacteria to do what they're meant to do in the device. But also allowing the exchange of information, let's say the drug, or different other molecules that might be interacting with the host. So these are some of the questions that we would like to understand what's happening at the interface.
Yeah, and it's a completely new frontier in material science that, of course, we are working on, and others here within the campus are working on, and also across the world. So whatever you figure out how to study the biocompatibility, biosafety of this is going to be extremely useful.
I hope so. I'm very excited with the topic.
And talking about inspiring things, that gets me to ask you about the favorite material that you've brought.
I'm glad you asked. So I was thinking also what kind of material could I bring here, because most of the things that I work with in the lab, are either sterile, I cannot bring them, or they have cells. I mean, I'm not gonna bring some cells here. So I brought my second favorite material that I use every day, and it's paper, actually. So I brought here my sketch block. I like to...
So can I...
Please go ahead.
Yeah, you can... Yeah. So the sketch block where, well, I'm aware, but I guess a lot of people... Is it fine if I open it?
Yeah. You can please go for it.
It's for those who are only listening, it's like an A4 size sketch block with some really nice kind of... what kind of paper is this? Do you know? Like it's sketching paper?
Tell us about it,
yeah.
Paper is actually made out of cellulose. Is one of the main components, which is polysaccharide that's present in the cell wall of plants.
Yeah.
So if you think of it, I mean, maybe there are some people that don't agree with me, but paper might just be like a dried hydrogel.
Yeah.
So because it's the cellulose paste that it's bound together with hydrogen bonds. So it's like a physical hydrogel, but they remove the water. So you can use it. So I actually brought a hydrogel.
That is a really fascinating take on paper, and it is the basis for like paper-based microfluidics, like the COVID tests and pregnancy tests and so on that you have.
Yes, correct.
But you, of course, use it for a different purpose, for making these beautiful sketches.
Yeah, I do like to draw.
From lectures and so on.
Yeah, so I like to draw a lot, but sometimes I don't have enough time to dedicate to this. So I kind of go to the lectures, trying to make some sketches, and I have a lot of fun with this. I try to gift them to the speakers when they look good enough.
Well, I have one of these, and I love them. It's right outside my office for everyone to see.
I'm so glad.
I'm very proud of it. And it's so amazing that you're able to do this within the lecture, and it basically becoms your lecture notes, but it's just much more pretty to look at.
It's a skill.
It is a skill. It is absolutely a
practice.
skill.
I improve every day, I think.
Yeah.
Paper. I also thought of paper because of the first episode with Roland that you did, that I listened to. So he works with these tactile materials.
Yeah.
So I thought, I choose paper also based on this. So the thickness of the paper, the roughness of the paper are good indicators for depending on what I want to use.
Yeah.
And I based most of my choices on these kind of things. And these are things that actually people, professional artists, look for. This the teeth of the paper, they're called. So these peaks and valleys.
Yeah, exactly.
And then thickness is another option. So depending on what kind of pen you're using or coloring method. That's what I brought today.
And it's really nice combination of science and art, which actually brings me to the last section of this podcast, which is about freedom.
Okay.
So, our Federal Ministry for Education and Research, the BMBF here in Germany, they've declared this year's Year of Science topic as freedom. So what is freedom for you in research and science?
Yeah, that's a really interesting topic. So freedom for me as a scientist, I think it's a right, but it's also a responsibility, right? So we are the ones that should keep track of what's the state of the art, and we are the only ones that can decide which science should be done. So we have to have this freedom, that's a right, to decide on these things. It's also a responsibility because it means that you need to report and you need to inform of whatever findings you have. I also think of freedom not as a, you can do whatever you want.
Yeah.
So it is always within a context, like in our research, right? So you look at, for example, in our case, we look at what the cells are doing in different environments.
Yeah.
And freedom, it's also within these environments and this context. So we need to keep track of the law. We need to keep track of the society and the government and the ethics as well.
Yeah, ethics is a big part, which, ethics can sometimes be something that seems to counterbalance freedom at times. But what is your thought on how that balance actually works?
I actually think it helps. So it provides like a backbone for us to be able to decide, because not everything is okay.
Yeah.
Right.
Yeah.
And even if it's a valid scientific question, maybe there's no way to test it because this puts in danger something greater.
Yeah, yeah.
And this is when ethics come in place to kind of course correct.
Yeah. So it kind of ensures the freedom of all
Yeah, exactly.
everything,
Yes.
basically,
Yeah.
like the idea of the ethics. And I guess it also probably plays a role in your own research when you talk about biosafety, biocompatibility. Ethics, I guess, is also part of that.
Definitely. So when we talk about biocompatibility and safety, we're talking about risks. We're talking about benefits. So this balance between what is okay, what level of risks are we confident taking on somebody else,
Yeah.
right? So these kind of questions, we have to deal with them
Yeah.
every day. Maybe not in the lab with our
Yeah.
small experiments, but on the bigger scale, of course, yeah.
Before we
definitely.
get to humans when we go
Yeah,
for
exactly.
these new therapies. Yeah, well, I think that's a wonderful point of view.
Awesome.
Take freedom and make sure that everyone has the freedom around it, even though we have the freedom to do science
Yeah.
in every way, but think of ethics and others
Yes, correct.
largely. So thanks a lot, Sara. This was a really wonderful conversation to have had with you, and I learned so much, and I got fooled real bad with
Yeah.
your statements, but I enjoyed it thoroughly.
Thank you so much for having me. It was an experience. I really enjoyed it. Thanks.
Wonderful. I hope it's the best podcast you've been on so far.
Yeah.
Well, you don't have to say anything. That's fine. Thank you, everyone, for watching and listening to the podcast. We had a great conversation with Sara Trujillo. And follow us on our social media channels, especially the group of the Leibniz-Institut for New Materials, INM. So we're there. We're there on Twitter, LinkedIn, Instagram, and a bunch of places. So look us up, and then you can see more content like this that we're going to be generating regularly. So thank you, everyone, and see you for the next episode.